you so much, Richard. Thank you to Anabios for organizing this conference and having me speak here today. And my lab is investigating the molecular mechanisms that regulate excitability within pain processing circuits of the spinal cord. And as we're doing this, we're using this translational approach where we're combining these rodent traditional kind of preclinical models with new human tissue assays that involve electrophysiology, but biochemistry and genetic approaches. And that's kind of what I'm going to really try to emphasize and unpack today, considering that's kind of the theme that we've heard of. And we've heard lots of great talks on pain. So thank you to all setting that all up. So I can jump right in and say the spinal cord is the middle sibling of the pain pathway. From Raj and Mike's talk, we have on one side, we have the periphery that detects and encodes those noxious stimuli. So you have transduction. And then on the other side, the spinal cord connects to the brain pain matrix that ultimately encodes that unpleasant sensory and emotional experience that we know of as pain. And so, yeah, peripheralists, I hadn't heard that term. So what's a spinal cord? Am I a centralist or a spinalist? And I think Raj is already converting over, and I think Ted, tomorrow, I saw a dorsal horn there, so we'll convert you to the dark side here of the spinal cord, because it's not telephone lines connecting the periphery where there's detection to the brain, where pain is in the brain, as we kind of all know and hear. In reality, the analogy of the spinal cord would look more like that. The outer layers of the dorsal horn of the spinal cord contain this complex neuronal network, this nociceptive network. And the output neurons from that network that are found in the outer lamina, lamina 1, yes, they receive inputs from those nociceptive afferents, both from the skin as well as the viscera, which we often forget. So thanks to that fabulous keynote of kind of unpacking some of that. But besides those inputs from the periphery, actually a ton of the input comes from local interneurons. Excitatory and inhibitory interneurons within that nociceptive network make up a majority, actually, of the input. And then there's also descending modulation from the brain in terms of efferents from the brain. And it's only after integrating all of that that you have signals going up. A good example of that in work physiology is the gait theory. What happens if you hit your thumb? What do you all do? You probably rub it. Or if a child falls, you rub their knee, because that activation of the mechanosensory circuits inhibits the nociceptive circuits. And so you have this finely tuned balance of excitability in physiological states. But in pathological states, as we heard Eric really nicely unpack some of the examples of that, you have that hyperexcitability. You can lose the balance between inhibition and excitation. And that's kind of where my lab lives at a molecular level, trying to unpack what's going on there and understanding that loss of proper regulation within those circuits that can drive pathological pain. And as we do that, we're using this kind of translational approach, as I alluded to, where we've heard lots of reasons of why sometimes we're not getting across, and we've heard our track record of not getting across. But when we look at it in terms of the context of pain, what do we have in terms of our tools for the preclinical models? Well, on this side, we often rely predominantly on male, at least historically. For physiology, which is where my lab does a lot of our work, it's often juvenile, just because of feasibility, and rodents. Whereas when we're thinking of the typical pain patient, for a lot of pain conditions at least, the prevalence is higher in females. It increases across aging, and obviously we have humans. And so one of the kind of untested assumptions, often at least untested, is that those molecular mechanisms and potential drug targets are conserved across this divide. But that's what we're setting out to investigate it, whether they're conserved across development, sex, and species. And today I'm going to highlight sex and species. I cut development for time so we get to those cocktails. So we're trying to bridge that divide. One of the targets that we're focused on, but I want to highlight, this is going to focus on NMDA receptors, but the kind of evidence I'm going to unpack, insert your target of choice, and we can use these same kind of approaches. But we're focused, one of the targets are these excitatory glutamate receptors, NMD receptors. And so they're critical for regulating excitability and plasticity at synapses throughout the nervous system, including in these dorsal horn nociceptive networks. And that's because when they're activated by glutamate and glycine, their agonist binding, they induce depolarization, so direct electrical signaling. But since they're calcium permeable, they also are involved in downstream calcium-dependent signaling that relates to changes in plasticity and gene expression, which we all know. One other critical detail here in terms of that's going to be relevant later on is a functional NMDA receptor is made up of 2 gluN1 subunits. There's one gene for that, but also 2 gluN2 subunits. There's 4 different genes for that. And the specific variants of gluN2 that are found determine the functional properties of NMDA receptors that vary, their regulation. And so that can be involved in tuning and the specific roles of those receptors. And you'll see why that's important in a little bit. But one of the first questions that we had was to look at how NMDA receptors are actually dysregulated in models of pathological pain. It's kind of central dogma that they're critical in central sensitization. We've known that for a long time, but I was actually surprised when I got into this field, we hadn't really looked at their functional properties to see how they're changed, at least in a lot of cases. And so that's one of the first questions that we are investigating, but across these variables, across sex and across species. And so I'm just going to do a one-slide summary of Anne-Marie. A lot of this is Anne-Marie Dedek, her Ph.D. thesis. She's in the audience. I'll have a picture of her to embarrass her further in a bit. But summarizing several years of research here in our inflammatory pain models and our neuropathic models, our basic male rodent models, we've identified this mechanism where we lose the brakes on excitability within these dorsal horn pain signaling neurons. And that's because you lose this chloride cotransporter, KCC2. It goes down, so you lose chloride-dependent inhibition, which subsequently causes the downregulation of this molecular brake, step 61, which is a phosphatase. So the main things to keep in mind are these are kind of brake pedal mechanisms, KCC2 and step, and they go down in response to BDNF that can be released from microglia or from afferents. So that loss of brakes, what we identified happens is it directly couples to an increased excitation. It causes BDNF, the receptor TrkB activates this Fin kinase, which then phosphorylates and increases the activation of a specific subtype of NMDA receptor, the gluN2B containing NMDA receptors. And so with patch clamp electrophysiology, we can record specific lamina 1 neurons that contain those out projection neurons within the dorsal horn. And what we see is in models of inflammatory pain or nerve injury, you see this robust potentiation of synaptic NMDA receptors. So loss of brakes directly couples to increased excitation at these synapses in male rats. And you probably know how I'm setting this up, where this is going. The other thing, actually, before I get to that, is if you take a spinal cord from a healthy rat and you just treat it in BDNF for like an hour, and we took this approach from Eve DeConnick's lab that had the kind of pioneering Nature papers on this, it induces the same pathway. So we can have an in-a-dish model that recapitulates what we see in vivo. And so that's in males. So here's the data from males, CFA, in vivo model, in a dish BDNF model, robust potentiation of these synaptic NMDA receptor responses. In females, no potentiation. And we have all the biochemistry and all the other experiments to back that up in rats. So from that diagram, that really busy diagram before mechanisms, there's many different targets. There's this whole mechanism, at least for males, that we potentially could target and test to see if it reverses pain. But in terms of translation, what about these mechanisms? Are they conserved as we move from rodents to humans? And so we have a unique ability, or not as unique now, it's emerging, I should say unique, but we have this ability to collect viable spinal cord tissue from human organ donors. We have at a hospital right next to us, next to the university, Dr. Eve Tsai is a neurosurgeon, but also a research scientist. So she collects the spinal cord tissue for spinal cord injury research, but we've been able to partner alongside her with that to also engage in this research. I should say right up front, we take that donation as a really selfless gift from both the donor and the family. So we really try to maximize the amount of science that we can do with this tissue, and we take responsibility with that. So what does it look like for these experiments? So Dr. Tsai is scrubbed into the OR after they've removed the organs for organ donation under these neuroprotective conditions, where the body is cooled, it's perfused with protective solution, and then within 1 to 3 hours of stopping the heart and those organ removals, the vertebral column can be opened and we can remove the spinal cord. And we, being Dr. Sai and actually Dr. Dedek, she's actually done this independently. Actually, Dr. Sai was away, and under a neurosurgery resident, she was able to do that. And I think that's part of the trick here, is being able to remove the cord under conditions where you preserve the viability, similar to all of that experience we have in the past from doing that with rodents, including adult rats, where the tissue is highly susceptible. And so that means we can collect the tissue into this protective solutions within 1 to 3 hours, and then do the same kind of experiments that we were doing with our rodents. We can do baseline physiology experiments just on naive treated tissue, or we can do saline versus this in-a-dish model of pain, BDNF, and do patch clamp electrophysiology, immunostaining against different targets, Western blotting to look at changes in targets, and I'll show you some of that data. I won't show you data where we've also collected tissue, flash frozen it for single cell RNA-seq studies that were conducted by Ariel Levine's lab. Emmanuel Bernet in France also was involved in, his team was involved in some of that collection. And so there's a paper that's now out, and it's a great resource for looking at some of your targets for human spinal cord. And we're already seeing examples of that. Ted's preprint had some great data from that as well, unpacking that data set. And Ted will obviously talk a lot more about this kind of stuff because he's the expert in leading a lot of this stuff. Okay, so in terms of the models, the in-a-dish model of pain that we saw for males. We see in human tissue, if you treat it with BDNF and compare it to adjacent spinal cord tissue from the same donor, so with its within group comparison, because most of these donors are not chronic pain. I should say that. This is an in-a-dish model. But what we see is that same loss of these inhibitory markers. So you lose the braking. The KCC2 and STEP go down, specifically at dorsal horn synapses, only at that fraction, not in the other fractions of tissue. Whereas the excitatory markers go up, FIN and GLUN2B go up, and not GLUN2A as a control. So you see that same coupling. That's in males. We see the same sexual dimorphism that we saw in rodents. For females, you wouldn't see these markers. I'm not showing that data, but I'm showing other data here conducted by Yves de Koning's lab looking at KCC2. It's that chloride cotransporter that gets internalized in males in response to BDNF or those in vivo rodent models. And so in humans, you see BDNF drives that internalization of KCC2 receptors at male synapses, but not in females. So you see the sexual dimorphism in this mechanism is conserved as we move from rodents to humans. So that caused us to take a step back and think, okay, like these are pretty striking sex differences. We need to go back and look at just baseline expression, baseline molecular and functional properties of these targets, these potential targets that we know are clinically relevant across these variables. And so that's what we've started to do. And so one approach to do that is with immunohistochemistry, staining, where we label the superficial dorsal horn, the pain processing region with CGRP, and then we do antigen retrieval to label, so we can look at all the NMD receptors, the GLUN2 subunits, including those found in the postsynaptic density. And this was started by a master student, Santina, and it was continued into the human tissue by Jennifer. And so what we see, and so this is good evidence of an example where the spinal cord is not the same as the brain, where there are these clinically relevant differences in that at brain synapses, GLUN2A dominates in the mature nervous system. Whereas we see here in the spinal cord, both for rodents and humans, yes, you have robust GLUN2A, but you have also robust GLUN2B and GLUN2D containing NMD receptors, which are a lot slower deactivating. And so there's a unique profile here, which is conserved across sex here, so male to females, for rats, and across species. And this is the ratio of SDH to DDH. So they're preferentially found in the superficial dorsal horn. So there's a unique kind of molecular fingerprint of these NMDA receptors in the spinal nociceptive circuits. But if you look at this, as you're staring at this, maybe some of you are already picking up, there seems to be a little bit of a difference. In the rats, you see it's more lateralized, the expression difference, where it's equal across that superficial dorsal horn medial lateral axis for humans. And that's relevant because we have somatotopic mapping in the rodents, right, which maps across for different regions of the body. And so we quantified that and found, sure enough, that was robustly seen. So this is the medial lateral axis, medial 0, laterals out here, and you see this robust lateralization, increased expression of these NMDA receptors in the lateral superficial dorsal horns that is not found in humans. So we do see this subtle species difference here at a molecular level. Okay, so that's molecular evidence of looking at the different NMDA receptors. We wanted to look at their actual functional receptor properties. And so to do that, we did these challenging experiments. And by we, once again, it's Anne-Marie. And so this was work she's continued on with as a postdoctoral fellow in the lab, funded by Eli Lilly and in partnership with Eli Lilly, where we're doing patch clamp recordings on lamina 1 neurons from rats, but also from viable human spinal cord sections. And so here's the painstaking data set from that. But initially, we were using criteria that were biased for those kind of moderate decaying NMDA receptors. GluN2A are the fastest, B are in the middle, D are the slowest. And so when we did this analysis, actually, this N should have been 4 for humans. This N should have been 10 here for male humans. So we were losing some with our analysis criteria. So we went back and kind of broadened our criteria to capture all of these synaptic events. And when we did that, what the surprising result that we found was there's this vast heterogeneity in synaptic responses within individual lamina 1 neurons that have various decay rates from synapse to synapse. So they're not homogeneous at all. So this is one neuron, representative neuron for each condition. And so what does this heterogeneity mean? Well, we know these decay rates correspond to different types of receptors. These are all excitatory miniature postsynaptic currents. The fastest being dominated by AMPA, then the next one's being dominated by those fast decaying NMDA receptors, GluN2A, then GluN2B, and then GluN2D. That's biophysic analysis. I've done at least recordings from lamina 1 neurons in rats to show they have these moderate decaying GluN2B dominated responses, some fast decaying GluN2A, some really slow decaying GluN2D. So we have evidence for that. So we went to our data set now and looked at the individual synaptic responses, because this was from populations. And what we saw was there's this fraction of all of these different types of events from synapse to synapse. So there's this intraneuronal heterogeneity in synaptic responses, which could mean there's this synapse-specific forms of plasticity, this additional layer of coding that we could have within these nociceptive neurons. And that heterogeneity is conserved as we move across sex and as we move across species. One really important thing that fell out of this, as you're probably staring at this, you might be picking up on, is that there's actually there's more fast GluN2A and less at least putative GluN2B-like events in human synapses. And we know some of those GluN2B targeting compounds failed in clinical trials. Maybe, this is really early days, this is preliminary, I should say, this is unpublished, so please, let's keep it. I'm sharing this early because I wanted to share where we're at, but this isn't out yet, but that could be relevant. In terms of, this is biophysics to correlate to the subtypes, we have proof of concept pharmacology, once again from a human lamina 1 neuron, looking at these different subtypes where we do baseline recording. Then we blocked GluN2B with row 25, 6981. The B-like events went down. We blocked D, the D-like events went down. We blocked A, and the A-like events went down, but not completely. This is a 2-hour recording from one neuron. So the amount of time we had so it's incomplete pharmacology, but at least it's great proof of concept that these different bins do correspond to those different molecular signatures. I wanted to unpack something that we're just starting to explore, and Anne-Marie is there as in her industrial postdoctoral fellowship with Lilly, where up till now we've been looking at molecules. And often we have targets, right? We look at them, and then we look at how manipulating those targets affects the behavior, affects pain. But what's happening within those nociceptive networks, those networks of spinal neurons that I first set up? And so we need tools to kind of understand what's happening, not at a cell-by-cell basis, but that kind of black box in the middle. And so we're starting to do multi-electrode array recordings on spinal cord tissue slices and high density. So we have 4,000 electrodes in this chip, so we can get a precise spatial map and temporal map of what's going on in time. And so we've been doing preliminary experiments both with rat tissue and with this human donor tissue. So it's the whole rat spinal cord or the dorsal horn of the human spinal cord. And looking at excitability, so individual spikes at different regions. And here's an example of spikes. We can look at changes in connectivity between those different putative neurons, or at least neuronal clusters. And then we can look at differences in types of waveforms between these. And that enables us to look at those over time now. We can look at populations. It also gives us more rich physiological data. Rather than patch clamp recording on one human neuron, we can really maximize what we can do with this tissue. And this was a video that, sorry, it doesn't play on a Mac, but you can envision those white spikes, dots, blinking in and out over time. How about I dance it? There we go. There. Over time. You can see the dorsal horn there. It's very captivating, and it's compiled over time. But I do at least show the output. I can show you the output of that. Keeping in mind this is preliminary data. We're still working with the manufacturer to get better spike detection through their AI. It was tuned for the brain. Once again, an example of how brain and spinal cord are not the same. It's not accurately picking out our spikes. So we're refining this. So we need to refine the analysis tools. But at least at a first approximation, we've looked at various agonists. We had BDNF, but it happened for these donors. CGRP, because they're female, we wanted, in terms of our burning questions, we want to see what CGRP does, because we've done a little bit with TED, kind of thinking about this. And so what you see here is it looks like CGRP. And once again, we can isolate the superficial dorsal horn. This is of a human spinal cord slice versus the deeper dorsal horn and look at spatial differences over time. So we have across space and across time. And we see here an increased excitability putatively induced by CGRP in the superficial dorsal horn. That's not as great in the deeper dorsal horn, which would be processing other somatosensory modalities. And here it's an N of 2. Once again, very preliminary. We're not hanging our hat on this, but this is just showing you the type of data we can get. And so with this kind of data, we can look at neuromodulators. How do they affect spinal cord pain processing circuits? We can look at potential drugs, like duloxetine was mentioned today. What does duloxetine do these networks? Gabapentin, whatever you want to think about, we can see what it's actually doing to these networks. And so that's kind of one of the exciting directions that we're going to be moving in as a team. And so I will close by thanking all of this work was not done by me. I have a fabulous team of dedicated scientists and a fabulous team of collaborators. So the members from my lab that were part of what I presented are in bold here. Lots of collaborators, including Jeff Krajewski's lab at Lilly, and Theron and many others at Lilly, collaborators at Adam Bios that have been involved in leveraging some of their assays in terms of calcium imaging to further test and unpack these so we can all move forward and drive the field forward as a collective group. Thank you so much for your time.
Thank you, Mike. Questions?
I have to ask, was the CGRP effect only observed in the female dorsal horn? Or
Too early.
don't
I
know
don't
yet?
think we've even had a male donor
Okay.
because we haven't had many donors. So, but for our NMDA, we're seeing CGRP seems to potentiate NMDA receptors in both males and females. And I think I've shared that data with you. So we're trying to unpack that. We don't know yet. We don't have enough data to get there. But
Cool,
that would be
thanks.
really curious either way, right?
Yeah, I agree.
Yeah. So that's our burning question there. Yeah.
Very nice talk. Thank you. So
Mike.
when you said superficial dorsal horn, it looked like it was above the substantia gelatinosa, if I was
So
eyeballing it correctly.
eyeballing, so that's another thing that we're refining. We do a screen picture afterwards, and we superimpose the electrode. So I think it's, from what I remember, I'm getting a nod from Anne-Marie because I asked her that exact question. So that's just a mapping logistics thing that we've got to refine further, type thing.
And
Yeah.
this might be going back in history a bit, but a while back, Pete Seberg and Michael Bennett looked at alternative splices in the NR2 subunit.
Yes.
Exon 5, exon 21, exon 22. And I was wondering if you had any idea if that affected some of the excitability that you see here.
We haven't looked at that. Those are great. Mike Salter's done a bit of, I think that was more of the GluN1 ones. And yeah, we definitely, and we're getting that RNA and we're doing some like qPCR, but we haven't done any kind of sequencing. So we'd be open to definitely.
Yeah, it's
Those are
sort of been laying fallow.
I
for a
know.
long
There's a
time.
lot of just low hanging fruit. There's lots to do, I'm sure, as we all can relate to.
The BDNF was really interesting because a while back we
Yeah.
showed in humans that had a hemideletion BDNF, they were very pain insensitive. So,
Yeah.
the lack of BDNF clearly has a profound effect on the sort of
Yeah.
nociceptive sensitivity.
Yeah.
Yeah.
Yeah. So it's definitely. And then did you look in sex? Male versus.
It would. I knew you were going to ask that. And no, there was no sex difference.
Okay.
I was
But that's
surprised.
the thing too. There's some nuances there. The trophic effects of BDNF.
Oh,
over time.
this is. I mean,
Yeah.
they grew up with only
Yeah.
half the dose. Yeah.
Yeah.
Thank you, Mike. And I want to thank, give a round of applause for all our speakers this afternoon, and that closed our first day.